So I'm just going to give you an overview of what we're doing in my lab. So we do a range of different things in my lab, and I kind of like to think of these as a sort of a spectrum. From one end, sort of very big data analysis, mining type of things, all the way to simulation and physical mining. And the sort of spectrum on one end, we're very interested in questions of biological knowledge representation, literature mining, text mining, and particularly interested in genomic privacy. Then, we get more into the sort of genomics and so forth, looking at just the variance in the genome, personal genomics, and so forth. And I'd say the center of the lab is on human genome annotation, and particularly disease genomics. From there we look a lot at, we're very interested in networks. Networks of genes, of course, but networks of proteins and more generally just networks of all sorts of molecules. And then the final level of zoom in, of course, is looking at those molecules in detail, and we're very interested in macromolecular structures. A lot of actually some new imaging projects on structures, and also simulation projects on structures and macromolecular motions. So that's the sort of overview of what we're interested in. And let me go now and just zoom in on some of the things. So I'll first talk about annotating the genome. You probably all know about the human genome. It's a very fundamental thing. We all carry copies of it, of course, in us. And we're very involved in annotating it. We've been involved in this for many years. Just on a high level, what this type of thing involves is there's different calculations that people do. Often one does an experiment on the genome and gets a signal of maybe its level of activity, its level of being bound, or its three-dimensional structure. So we do a lot of signal processing to kind of smooth these signals and chunk them up into little regions. And then we also group these in regions into larger annotation blocks and networks. Another type of genomic annotation is just looking at parts of the genome that are conserved. But this actually is a somewhat complicated thing. You can see the conservation within the human population between two people, between human and mouse, or even between different parts of the genome. And so there's a lot of genome annotation that looks at kind of repeated parts of the genome. So I'll go to the next slide. And this slide just kind of looks a little bit in more detail at this kind of conservation type of analysis. We're very interested in pseudogenes and genome remodeling, which is a particular type of genomic conservation where often a gene is duplicated. Can you guys see my mouse? I just want to make sure you can see my mouse bouncing
Yes.
around. Yeah, you can see the mouse. Yeah, so where a little bit of the genome is duplicated, and then often that duplicated copy dies. And that's kind of interesting. It sort of gives a lot about the history of the genome and what it was like in the past and what's died out now. It's kind of like looking at, well, essentially it's molecular fossils. And these fossils are created by different processes, just like the fossils that we have in the ground, from animals. And some of the two main processes that create them are obviously duplication and death, but there's another process which is retrotransposition, which involves the insertion of many different elements of the genome. And we look at this, and turns out in our molecular history, there's been a number of very big retrotranspositional explosions. And we're kind of interested in that in terms of genomic history. So here's a little bit on networks. So now we've kind of talked a little bit about the notion of annotating the genome. So the next thing we can do is kind of connect these elements together and make networks. And here's a picture of actually kind of the regulatory network for the human genome. This is the network of transcription factors. Now it turns out that one thing can regulate something else, but it can also regulate another regulator, and this leads to some very complicated networks. And this picture kind of shows a little bit of the sort of tangled hairball that you tend to get when you start to look at these molecular networks. And analyzing these hairballs, there's a lot of interesting things you can do. You can find parts of the network that are very connected, parts that act as bottlenecks, parts that act as master regulators, and so forth. And this is a kind of, there's also an open field of network science and analyzing networks. Let me go to the next one here. So we do a lot of machine learning models for genome annotation and trying to model the effect of variance or model the effect of different annotations in the genome. We've done this obviously for a very long time, and this just shows a very early slide where we were trying to predict the output of the genome, the expression of the genome, from the kind of upstream chromatin, or how open the promoters and enhancers were in the genome. And it just shows a little bit of some of the inputs to a machine learning model that we had worked on for this. This slide gives a little bit more onto the personal genomics and looking at the variance in the genome. Now, you're probably all familiar with mutations, SNPs. Those are base changes in the genome. And it turns out we have a lot of other types of variants in the genome that are not as well understood. These are called structural variants, and they're often things like deletions or insertions in the genome. And there's a variety of different techniques people have for finding these. They're a bit more complicated than finding single nucleotide polymorphisms. And so, for instance, here, let me just show you the kind of easiest one, where you can kind of imagine there's a deletion in someone's genome compared to, say, the reference. You can sort of see that by looking at a read from the person and allowing that to map in a different way, where it maps in a kind of split pattern. There's another technique that you kind of look at the pile up of the reads in the genome, and you get a kind of signal, and you can process that signal to find deletions and so forth. Now, a big part of annotating the genome is to kind of put these things together, put the variants together with the annotation, and try to understand which variants in the genome are going to have a big impact, obviously in terms of causing disease, but also in terms of causing just phenotypes in general. We've been very involved in this variance interpretation type of thing. And this just shows a particular scheme from a paper a number of years ago, a very simple scheme that's easy to understand, where we would annotate variants in the genome. This particular one basically looked at, do the non-coding variants sit in annotation? Are they in regions that are conserved? Do they have some obvious mechanistic impact in terms of breaking a motif and so forth? How do they affect the network that this annotation sits into and so forth? And this was quite useful for annotating. This particular scheme was actually very useful for annotating what's called somatic variants, which are not germline variants, but somatic variants that occur in cancer. And we've been very involved in, in addition to cancer, and looking at neurogenomics. We're very interested in things to do with the brain. And recently we've built a number of regulatory networks and machine learning models associated with variants in the brain. This shows a particular type of a machine learning model. It's a deep learning model that tries to go from the variants to predict various neuropsychiatric diseases such as schizophrenia, autism, bipolar, and so forth. And one of the neat things about this model is that it tries to be an interpretable machine learning model. So the connections in the model actually are taken directly from the physical gene regulatory network, so that you can interpret the connections a little bit better than you can in these models in general. And so this is a particular type of model. And here's the connections that are actually highlighted in particular predictions often suggest, because the model is interpretable, suggest particular target genes that are involved in, say, one of these phenotypes, such as schizophrenia. And that's kind of useful in terms of targeting these genes for drugs. In this particular model, it was quite a big thing. We had a paper in Science that came out in this whole package that was sort of focused on the brain and so forth. Now, I'll just tell you about a final area here, and that's looking at genomic privacy. One of the main barriers to kind of really large-scale interpretation of the genome and a lot of biomedical data, of course, is privacy. In particular, our genome is very private to us. It tells a lot about us, and a lot of other biomedical data is as well. And we have to think about potential privacy leaks that are not obvious, and also how to surmount them. And so this picture here kind of diagrams a particular type of attack that you can make on the genome. It's called the linking attack. I won't go into great detail on it, but the basic idea is that you can use the genome in a very non-obvious way to kind of create a cryptic quasi-identifier for a person, and then look them up in a public database, and then perhaps fetch sensitive information about them, such as their HIV status and so forth, from this kind of cryptic lookup. And then the final bit I'll talk a little bit about is about simulation and structures. So obviously a goal of a lot of these things is really to find specific molecules that are associated with disease. And then, of course, the next goal is to make a small molecule or a pharmaceutical that's going to affect these molecules. And so to do that, you really want to think about where a small molecule would bind on a larger molecule and how that binding is going to be affected by variance in the molecule. And so this just shows a picture of an actual molecule. It's a very complicated surface, and it sort of diagrams, so it sort of schematizes a little bit of the Monte Carlo or simulation type calculations we had to kind of roll a ball around the surface to find places that were particularly sensitive to variance, and maybe were very sensitive to where a drug would bind. And just to sort of wrap it up, we're also very interested in kind of perspective. We've been doing this biomedical data science, or obviously for a long time before it was very cool, and so we're very interested in kind of perspectives on the field, and we're very interested in kind of thinking about issues of the scaling of the data growth, the different types of data modalities, and how the algorithms are in a sense scaling with the data growth. So that's a kind of overview, and I have just a little quick summary here of some of the topics again that we're into. Again, there's the knowledge representation, literature mining, personal genomics, looking at the variance, genomic privacy, the human genome annotation, a lot of the machine learning models, especially the interpretable ones, comparative genomics, pseudogenes, the disease genomics, a lot of interest in cancer genomics, neurogenomics, variant interpretation, just a lot on network science, networks of genes, macromolecular structures and motions, and so forth. And we're also, I'd say, we're getting into some new directions now with, sort of digital phenotyping, using wearable devices, and the structure stuff. There's a little bit of image processing and so forth associated with that we're very into now. And with that, I think I'll end. Thank you very much for your attention.